Yes. First, a comment. I would say that a lot of people would think the sarin attacks by the oil were fairly successful. But that said, the reason that the anthrax attacks did not work is because the strain involved was the Stern strain, which is a non-virulent vaccine strain. And that's what they had, and that's what was collected. Now, they had a building that they rented, and they had people in that building, and they were spraying the stuff out of the rooftops. That's why people complain, it smelled.
Right.
And so they obviously knew that it wasn't that dangerous, because they wouldn't have occupied the building that they're spraying a pathogen out of.
Right.
So nobody's really quite figured out why they were doing what they're doing, if somebody sabotaged their system, or if they were still in the process of testing, because if you're spraying it out of a building, first of all, you're using live culture and not too many spores. But you could put out plates, for example, in an area, and the colonies are quite specific, so you could figure out how far it went.
Right.
They may have still been done testing. So I'm not sure that you can say that it failed because it was doomed to failure because it's Stern strain, which is not infectious. Now, I agree with you for the most part, but for example, you're talking about human pathogens, but a bio-threat attack might not be against a human.
Right. It might
So
be
for example, let me give you an example of rice.
Okay.
You want to destabilize a country, take down their stable food product.
Right.
So rice, for example, is resistant or is sensitive to a lot of fungi.
Right.
And the way they treat that is, for example, to use copper-based fungicides to knock down the metals, and those are very commonly used. And too much copper can be a real problem, so you can't up the copper too much above a certain level. Now, it's been known since the 80s why fungi are resistant to copper. It's because they overproduce an enzyme a protein called metallothionein. And that's been studied, that's been all that work's been done. And so if you could engineer copper resistance into some of these really nasty plant pathogens, you could have a real problem, and you could work with that in the lab, and you're not going to kill anybody.
Right.
So if you're looking at human pathogens, I agree with you. Actually, I agree with you a lot of this because all the different steps are really difficult to pull off. I completely agree with that. But you could pick something where you could make a simple change and do something really nasty. Now again, you got to grow the stuff up and all those different steps, you know. And I've done all these sorts of experiments, and I know how hard it is to do. And nobody's just going to pull it off overnight. But a state program could do it.
Right. Okay. I didn't focus on that part of it.
Yeah, but in any case, for example, the metallothionein thing, I could do that
Probably, yeah.
in one of these labs.
It's not that hard to engineer copper resistance.
On the other hand, getting drug resistance, you don't need to do all this, all you do is selections.
Right, mutagenesis.
You don't need to do mutagenesis. You actually just do selections. For example, we had about 520 isolates of bacillus Stern that were selected for resistance to Cipro, which is supposedly the drug of choice for anthrax, even though I myself I take penicillin because you can't seem to get resistant to that, or doxycycline.
I've taken that.
And it was just you start out with a concentration that you know will, basically at the margin, a few will survive, and you select for the survivals. You then increase the concentration of the drug, do this over and over again, and you get resistance. And you can get resistance to very, very high levels of those drugs.
Sure.
And you don't have to do any of it. It's just selection. Now, if you do too many rounds, you tend to lose pathogenicity. That's another issue. So, anyway, I'll leave it at that. I'm sure there's lots of questions.
Thank you. Yes.
I had a question. So first of all, thank you very much for this very interesting presentation. I was wondering if the first three steps, if considering a black market, for example, because of new technologies like labs getting new stuff, what happens to the old equipment? What do you think? What is your assessment? How likely it could be for kind of a sophisticated biohacker to get some equipment because of the black market opportunities?
I think any equipment that any laboratory equipment that is needed to build a bioweapon can currently be obtained through the open market, through eBay, and and I'm not aware of any restrictions on laboratory equipment per se, perhaps on strains, perhaps on reagents, but with respect to the laboratory equipment, I don't think the black market would help much because the open market is already so accessible. Okay. With respect to strains, which I think is the most dangerous of these steps, yes, if if a black market were to exist and a biohacker were to able to get his hands on a pathogenic strain, that would be a problem, but I'm at least not aware of any black markets that exist to sell these strains. Yes.
You define biohacker in a very restricted way. Basically, undergraduates or recent graduates who are go out having a good time,
Not as biohackers,
break,
right?
break into the Defense Department, not to launch a nuclear weapon, but because they can
But
tell their
for
friends
frap-
that they broke into the Defense Department system. Somehow, well, I agree with your general sense for, you know, for that very narrow group. That's not really the very narrow group, the group that I'm worried about. I'm much more worried about his comments about Shinrikyo, or people where issues of access to product, cost, laboratory space are irrelevant.
Right.
It's way, you know, $50,000,
It's not.
you know,
Right.
is pocket change. So it's those other issues, the CRISPR-Cas9 ability to turn camel pox into smallpox, for example, which probably 2 people in this room could do. You know, that's the biohacking that concerns me, especially among disaffected people. So if you have a postdoc at Duke who is really pissed and has the equipment around him or her, it's a disaffected biohacker with intent.
Right.
Much of the things that you're concerned about are irrelevant.
Right. I obviously didn't focus on that kind of group. I'm an undergraduate. I don't know what a postdoc would know, so I felt that I was unqualified to comment on them. But to my understanding, most biohackers are within the group that I described, and I guess my conclusion is that the majority of biohackers, the enthusiasts currently operating, are not threats. But I agree, if a disaffected postdoc were to want to make one of these. I've spoken to a professor, for example, after this was published, who said, I agree with you, my undergrads probably couldn't make a bioweapon, but I can. So, but we hope that we can pay our postdocs enough and our professors enough so that they're not disaffected.
The best solution.
Right.
One question here.
Oh, yes.
First of all, comment on that. I did biology all day, you know, sometimes 16 hours a day. I didn't want to go home and do it as a hobby, thank you. But, and I know how to make a bioweapon. But what about, rather than saying you're going to have regulations, why not monitor what's being purchased, for example? Don't have a regulation, don't have restrictive regulations, but just for example, DNA sequences. Why not have a national system that monitors all of it, and you can computerize that, pick out things that people want to look at. So for example, I used to work on castor beans and genetics because that's what produces ricin. And I ordered 500 grams of castor bean seeds. And no problems, I ordered it. And about a month later, I went to a meeting with the FBI. One of the guys comes up and says, I didn't know you were working on castor. And I said, how did you know that? Well, the guy that supplied the beans called the FBI and said, some guy just bought a pound of castor beans. That was voluntary. But why not have a system where it's reported? It's not restricted necessarily, but just reported.
I think that's almost similar to what's happening with genetic DNA, for example. I tried to look into this. I'm still not clear whether the guidance on synthetic DNA is done by the government or just by self-regulation agreement between different.
I don't think there's a government rule about that.
Yeah.
I don't know for sure, but I don't think there is.
I have sort of a comment on that. I'm Robert Shaw. I direct the export control and non-proliferation program here at CNS, and I have looked into the screening guidance that's used by suppliers of synthetic DNA as sort of what in the export control world we would call sort of red flag guidance to detect signs of a suspicious purchase or inquiry. And it's of interest in the export control domain because it's one of the most well-developed red flag detection frameworks that's out there. Other industries, it doesn't even compare to what you would find in the synthetic DNA industry, which is quite impressive. And my impression is that much of this was sort of generated within the biotech industry. In other words, the screening guidance largely came out of industry associations, discussions, perhaps out of concern that a regulatory regime might appear that would not be, well calibrated to the actual workings of the business. So it does seem to be somewhat, at least, or kind of call it self-organizing or self-regulatory.
Right.
So that was a comment that I had, and I think kind of broadly though, I was, but in a similar sense, you had suggested one of the recommendations you had with regards to the purchasing of laboratory equipment and regulation there is that potentially the community labs might develop some sort of licensing arrangement in which they would sort of license purchases of this equipment, almost from the purchasing side, which is interesting. And I was just curious what your thoughts are on the appetite for such a licensing arrangement within those communities. If you look at the synthetic DNA guidance, again, this is more on the supply side. It happened. I mean, there was some, clearly it was sort of self-regulatory. There was some interest in doing this, so maybe something could happen here. But I thought it was quite interesting. I was just curious if you had a sense of the appetite for this, or whether there's already discussions that are kind of bubbling up within the biohacking community.
I mean, my exposure to that is very minor. After I published in the bulletin, I did get some comments both on the article and through email from unhappy biohackers who did not like it at all. Of course, this is likely a very small. I hope it's a small subset because honestly, to me, regulating asking people to participate in community biolab does not sound that restrictive. But their argument was that the whole point of DIY biology, the entire movement, is to democratize synthetic biology so that everyone could have access to it and contribute to the innovation. And to them, the restriction of laboratory equipment in any ways doesn't seem to be very appetite. Right.
Okay. I want to make one note, and that is about when you went through the different steps that needed. For example, the first one was that you needed, if you're going to work with dangerous pathogens, you're going to have all the equipment and all that.
Sure.
But my experience as an inspector in Iraq, we inspected, for example, Al-Hakam, and at the first we were thinking, oh, there's no safety equipment at all. The dryers and all that are out in the open. They're not working on pathogens, but that turned out to be not complete at all, because all they did was put on a gas mask and worked perfectly happily with bacillus anthracis and botulinum toxin. So you can't, for other countries, you can't put the same standard on them as we have here. You got to keep that in mind.
I wasn't aware of that.
Yeah.
Along those lines, Kazakhstan, same sort of thing. You go there, and I went into the facility and they gave me what they normally wear, and I'm thinking, I don't get paid enough to be here. And it's what they do is they do with procedure what we do with engineering tools. And the way they have just as few accidents, or maybe even fewer than we have. And the way they do that is they train these people and they look for certain characteristics. They don't want anybody to take any shortcuts, among other things. But once they're in there, they do it by procedure rather than by all the equipment. And they can't afford the equipment, and a lot of these third world countries can't afford that equipment. In fact, in India, we were talking about exactly that last year, where they say, well, we want a program like the U.S. Well, the program for safety by the U.S. is way too expensive.
I see.
And you can train these people, they handle it, and people handled it for 100 years without all this equipment safely. So it can be done. Now, that said, it's nice to have the equipment. On the other hand, if you've ever had to wear the equipment, it's no fun to wear it either. I mean, even at level 3, it's no picnic. You get into all that equipment, the first thing you have to do is go to the bathroom, and you got to get back out of it. And it's no fun at all to use the equipment.
I've been lucky to only work at level 2.
Well, I think this is very good, and I think learned a lot, and we'll have you again when I teach my course.
Thank you so much.